Okay, this is on the life cycle of the erythrocyte. So as you know, blood cells are a little bit different. Red blood cells are a little bit different in that they're essentially just masses of hemoglobin. So they, as a reticulocyte, they get their nucleus ejected, so they, they can't go through mitosis. They're not like other cells in our body. So the question is, what happens to them after they die? So think of a red blood cell as essentially just masses of hemoglobin, and then hemoglobin is going to be made up of two parts, heme and globin. So let me go to the next slide, and then I'll bring this slide back. So an erythrocyte is a biconcave disc. That's just meaning that, it's just the shape of it. Don't worry too much about that, but as I was saying, it's essentially masses of hemoglobin made in the red bone marrow, and its life cycle is about 4 months. After that, it's phagocytized, eaten up by the liver and the spleen. So let's go back to hemoglobin. What is hemoglobin made of? Well, the name hemoglobin. All right, so it's made from heme and it's made from globin. Globin is just a protein. So whenever you see globin in a word, or glob, G-L-O-B in a word, you're going to think protein. All right, so we have the globin, and then we have heme. Heme is a iron-based pigment, or I shouldn't say that. It's a pigment with an iron component. Right? So if we were to look at what hemoglobin is made of, it's made of three things. It's made of amino acids, which is the protein part. It's made of the pigment, and it's made of iron. Right? And so we've got about 280, or I wrote 300 million hemoglobin molecules per red blood cell. Right? So when we look at what's going to happen when these erythrocytes get phagocytized at the end of their term, we're going to look at three parts. We're going to look at the iron, the pigment part of heme, and we're going to look at the amino acids. So globin's a protein. This part's really easy. Proteins are, in general, recycled, and they're broken down into amino acids, and they're rebuilt back up into other proteins. Right? That's just anywhere around your body. Or if you think about the protein you eat, that's what happens with it. It's broken down into amino acids, rebuilt back into proteins. So this is pretty easy. Globin is broken down into amino acids and reused. Right? It's mostly, in this case, reused to make more globin, but theoretically it could be reused for anything. So the globin part's easy. Globin's recycled. Right? So now we're left with the heme part. And so we've broken the heme into an iron part and a pigment part. So let's deal with the iron part first. Right? So the iron is recycled. So we have the globin protein being recycled. We have iron being recycled. Right? So what happens is that transferrin is a protein, trans meaning to transport. F-E-R-R is going to tell you that it has something to do with iron. So if you see that in a word, it has something to do with iron. So transferrin is going to pick it up. Transferrin is going to pick up iron and transport it to a storage protein. It's one of the two storage proteins. This is the main one. So the main storage protein is ferritin. Right, so transferrin picks up iron. It's going to store it as ferritin. And don't forget, you're eating iron as well. So you're getting iron from your food. That's also getting stored in ferritin. So after a while, after you get some iron stored up, then transferrin's going to pick the whole thing up and take it over to the red bone marrow, because that's where they're made. That's where red blood cells are made. So, again, transferrin picks it up from the liver, from spleen. It's going to hold it for a while in the protein storage ferritin, and then transferrin is going to pick it back up and take it to the red bone marrow. All right, let's look at the pigment portion. This is the most difficult part, and this is not recycled, right? The pigment is excreted. So there's a few steps to it, and the first is that we have biliverdin. And so as that VERD suggests, it's a greenish pigment and from there we end up with bilirubin. Out of all these pigments, bilirubin is probably the one you're going to come across in lab tests. For example, if you have an elevated level of bilirubin, it could be indicative of hemolysis. Your red blood cells are breaking down more than we expect them to be, right? And in any event, this is the yellowish pigment that is going to be used in bile. And so you might be thinking the spleen is breaking down red blood cells as well. Well, in that case, the bilirubin is going to be carried by albumin. If you remember, that's one of your plasma proteins. All right, so albumin is going to carry any other bilirubin over to the liver, right? And so at this point, it's going to end up, bilirubin is going to end up, since it's in the bile, it's going to end up in the intestine, small intestine, then large intestine, right? So in the large intestine, bacteria are going to break up this bilirubin and turn it into another pigment called urobilinogen. And then urobilinogen is going to go through one more degradation. It's going to turn into stercobilin, and that's going to give color, a typical color to your feces. And then on the other end, some of that bilirubin is going to be processed by the kidneys, and it will be degraded into urobilin, and that's what gives the urine like the yellow color. So let's go back again to this whole chart here. So here's the red blood cell. It's getting broken up. It's getting phagocytized by the liver and the spleen. It's essentially, red blood cells are essentially hemoglobin. Remember, hemoglobin is made up from heme and globin. Here's the globin. So let's follow this up. Globin gets recycled. The amino acids gets broken down into amino acids, and it gets recycled. All right, so that part's easy. Let's go to the heme part. All right, so here's the heme part of the hemoglobin. Heme has a pigment component and an iron component, right? So again, iron gets picked up by transferrin, stored in ferritin, and that's what this is kind of saying here. And then transferrin picks it up again, takes it to the red bone marrow to be used again to make new erythrocytes. Here is the heme part, the pigment part of heme that gets excreted, right? And what you don't see in this blue line that I'm tracing is all of the names of the pigments, but you obviously still need to know them, right? So biliverdin, bilirubin, urobilinogen, stercobilin, and urobilin. So that's essentially what happens to red blood cells. Two parts get recycled again, the iron and the globin. The pigment portion does not get recycled.